Hello, everybody. Thank you for joining us for our Precisionary webinar today. It's March 7th in the year 2023. And this morning, we are honored to welcome Dr. Junshi Wang, who is an instructor in the Department of Neuroscience at Mount Sinai. Today, Dr. Wang is going to tell us about his use of calcium imaging in electrophysiology to explore the interaction between neurons and astrocytes and the condition of addiction. So Dr. Wang is an enthusiastic neuroscientist who is joined by, I think, an enthusiastic crowd today. And he has been investigating neural mechanisms underlying addiction for, 14 years. He earned his PhD in neuroscience from Arizona State University. And while there, he used behavioral models in immunohistochemistry to investigate how BDNF, or brain-derived neurotrophic factor, signaling in the brain reward circuit helps to regulate stress-exacerbated addictive behaviors. In his first postdoc, he was in Dr. Yan Dong's lab at the University of Pittsburgh, and there he learned electrophysiology. And being somebody who is interested in tinkering and engineering, he has been using ETHIS since to study neuroplasticity in addiction. Currently, he is a postdoc in Dr. Paul Kenny's lab at Mount Sinai School of Medicine. And there he enjoys making modifications to procedures and instruments, including our compressed-dome vibrating microtome, to gain better control at each experimental step, ultimately leading to better quality data. And here today, he is going to share some of those modifications, experiments, and experimental results with us. So with that, Dr. Wang, I want to hand it over to you and let me make you a co-host so you can share.
All right.
All right. So Dr. Wang, we see your presenter view. If you want to swap it, that is wonderful. Perfect now.
All right. Okay.
We can see everything perfectly.
Okay. Yeah, like Abby introduced, my name is Junshi Wang. Right now, I'm an instructor at the Icahn School of Medicine at Mount Sinai. And today I would like to share my research on synaptic plasticity and how cocaine via astrocyte triggers synaptogenesis in adult animals. So during graduate school at Arizona State, I focus on the function of a brain-derived neurotrophic factor in drug addiction. On the red panel is some immunohistochemistry labeling of BDNF in the ventral tegmental area. And then I received postdoc training at the University of Pittsburgh from Dr. Yan Dong. There I completed a series of studies investigating the synaptic mechanism underlying addiction, depression, neuropathic pain. Then I joined Dr. Paul Kenny's lab at Icahn School of Medicine at Mount Sinai. Currently, I'm using single-cell RNA sequencing, spatial transcriptomics to study the function of a genetically defined neural circuit in addiction. So what exactly do I study all these years? If you want me to summarize in just one word, it will be synapse. I'm fascinated by the information processing and the computation happen in the synapses. I study how synaptic changes in a circuitry or in a genetically defined population of neurons regulate distinctive aspect of addiction-related behaviors. So our view about the function of the synapse has been evolving at an early stage. When the synapses were first identified using neuromuscular junction, the field views synapse as simple connector, which only transmit the information among neurons. The molecular complexity and the computational properties of the synapses still await to be discovered. Later on, the synaptic strength model was raised. In this model, it suggests that the increased efficacy of synaptic transmission strengthens the firing in the circuitry and facilitating certain learner behavior. At this stage, they use hippocampal synapses as the study model. The neuroscientists realized the full potential and the beauty of the synapses. From their studies, we learned that synapses can compute the information in patterns of the neural activity to generate a specific behavior response. And the landmark discovery during this period of time was the finding of a long-term potentiation, which launched the field of synaptic plasticity. And during that stage, there is a hot debate about the expression site of the LTP, whether it resides in the postsynaptic site or due to presynaptic release change. I want to highlight those debates. First, those debates leads to the discovery of the silent synapse, which is the focus of my study. Another reason I want to highlight those debates is they carry a very valuable lesson. So as you can see, the first paper on the top, they say that LTP involves presynaptic release change. The second paper said, no, it doesn't require any presynaptic release change. Then finally, the third paper reconcile and interpret the discrepancy between those two studies. The reason the first paper say the LTP involve presynaptic change, because they use the coefficient of a variation and the failure rate of synaptic responses as the readout for presynaptic release probability change. When studying the neuromuscular junction, those two indicators can accurately reflect presynaptic release change. However, for hippocampal synapses, which are more sophisticated, those two indicators cannot exclusively indicate presynaptic release change. For example, the emergence of a patch of a functional AMPA receptor at a postsynaptic sites will significantly alter the CV and reduce the synaptic failure rate. For example, here, as you can see in this panel, each dot represents the amplitude of a synaptic response. They gradually reduce the stimulation intensity till the intensity is too weak to consistently induce any response. And when you hold the neuron at -60 millivolts, there is no synaptic response. However, if you depolarize, you increase the holding memory potential, you can see synaptic response gradually showed up. And also, if you apply DAP5, a blocker of NMDA receptor, you can block that synaptic response, suggested those response is mediated by NMDA receptor. So the discrepancy of failure rates between -60 millivolt and +30 millivolt and its dependence on NMDA receptor indicated that there are synapses containing only NMDA receptor, but no AMPA receptor. This 2022 Nature paper elegantly illustrated the structure substrate for silent synapses in adult neocortex. The left panel shows the distinctive structure between regular spines and the Filopodia. Filopodia, as you can see, defined as the protrusions lacking the distinct head. And they account for 30% of the total number of dendritic protrusions in the cortex. Also, their immunohistochemistry study show that many Filopodia, although they have presynaptic partner nearby, they have NMDA receptor, but they don't have any glutamate receptor. They don't have any AMPA receptor. So the red panel, they use 2-photon NKG electrophysiology to demonstrate those Filopodia, they don't have AMPA receptor. If they NKG the glutamate near a regular synapse, you can induce a clear response. However, if you NKG near the tip of the Filopodia in the light blue trace, there is no response. And if you put the neurons in the magnesium-free environment, which remove the magnesium blocking in the NMDA receptor, even you NKG near the Filopodia, you can induce the synaptic response. So this data elegantly demonstrated that Filopodia only has NMDA receptor, does not have AMPA receptor, and they can be the structure substrate for silent synapses in adult animals. So right now we know the existence of silent synapses in the brain. Why we should care about them? Why they're important? The main reason silent synapse is important is they will turn into regular synapses. They will not keep silent forever. Functional connectivity in adult brain is very flexible. And the silent synapse on Filopodia can be unsilenced through Hebbian plasticity by recruiting new active connections in circuitry. So the panel show that after the LTP pairing, there is a significant increase in the synaptic efficacy. So memory formation actually requires a balance between flexibility and stability. Flexibility establishes the acquisition of the memory, and the stability is necessary for the long-term memory retention. In this case, as you can see, the panel on the right, after the LTP protocol, the Filopodia turns into regular synapses, and they can mediate synaptic transmission. Because our study mostly focused on drug addiction, so we hypothesized that maybe the initial exposure to drug-induced silent synapse, they are flexible synaptic plasticity, and upon repeated exposure, those flexible synaptic plasticity turns into a long-term stable connectivity change. And those connectivity change can carry the drug memory, which drives the compulsive drug taking and chronic relapse. So natural rewards activate the reward system and trigger dopamine release into the nuclear accumbens, which are circled by the red dashed line here. This region is also known as the brain reward center. And the dopamine release into this region, the nuclear accumbens, lead to euphoric feeling and reinforcing the behavior that triggers the dopamine release. So this reinforcement is a key learning component because it motivates behavior that is supposed to benefit survival and well-being. However, drug abuse hijacked this system to prioritize the drug taking and drug seeking over anything else. Our hypothesis is silent synapse could play a crucial role in such drug-induced behavior adaptations. Therefore, we decided to study the silent synapse in the nucleus accumbens, because once we fully understand the function of silent synapse and the mechanism underlying their generation, maturation, we could treat drug addiction by targeting silent synapse. So to do this, I use self-administration model in rodents. So first, we implant a catheter into the jugular vein of either mice or rats. And after a week of recovery from the surgery, they were exposed, they were trained to press the lever to obtain intravenous cocaine infusion. And only pressing the active lever will trigger the cocaine infusion and turn on the Q light. The Q light will serve as a conditional stimulus. So after the animal acquires the self-administering behavior, we can put them through forced abstinence. Then we can measure the drug-seeking behavior triggered by the Q lights at a different withdrawal time point, which can be used to gauge the drug-seeking and the propensity of relapse. I use BII lipophilic fluorescent dye to stain the dendritic spines, and I quantify the density change, especially of the Filopodia. My results show that on withdrawal day 1, cocaine exposure significantly increased Filopodia density in the medial spiny neurons in the nuclear accumbens. So which suggests that cocaine could trigger the generation of silent synapse in adult rodents. So now raises another question, how we could functionally quantify the ratio of silent synapse, especially in the circuit of interest. I take my paper published on pain to illustrate how we quantify silent synapse in a pathway-specific manner. So here, each dot represents the amplitude of the synaptic response, and the traces show you the response and the failure when the neuron held at -70 millivolt. And the traces above show you the failure rate at +40 millivolt. So to gauge the relative level of silent synapse using electrophysiology, we focus on the discrepancy of a synaptic failure rate between -70 and +40. First, we hold the neuron at the -70. We use electrical stimulation to evoke the response. Then we gradually reduce the stimulation intensity till the response becomes inconsistent, which means the failure rate reach around 30 to 70%. Once the failure rate stabilizes, we continue recording for 10 minutes. Then we change the holding potential from -70 to +40 millivolt, and we keep recording the failure rate. After we recorded the failure rate at a different holding membrane potential, we can use that equation to calculate the relative ratio of the silent synapse. In essence, the larger the differences between the larger the differences of the failure rate between that two holding potential, the higher the number of silent synapses in the recorded neuron in response to the treatment. Now back to the original question about how to measure silent synapse in a specific circuitry. We take advantage of pathway-specific optogenetics. So we express the channel rhodopsin in the neurons in the media thalamus. And we study their synaptic connection within the anterior cingulate cortex, ACC. As you can see, the channel rhodopsin expressed in the soma, in the MD. And there's a lot of fiber in the layer 2, 3, and layer 5 in ACC. So we first use the optostimulation to trigger synaptic response. Then we gradually reduce the laser intensity till we achieve the optimal failure rate when the neuron is held at -70 millivolt. Then we gradually elevate it till +40 millivolt and record the failure rate at that level. Then we can calculate the ratio of silent synapse. My results show that neuroplastic pain significantly increased the level of silent synapse in the MD to ACC circuit. Importantly, by interfering the generation of silent synapse in this particular circuit, we ameliorated the allodynia caused by neuroplastic pain. So now back to the cocaine addiction question. We measure the level of the silent synapse in response to different duration of cocaine exposure. Consistent with my spine morphology result, 5-day cocaine exposure increased the level of silent synapse in the nuclear accumbens. Now we're wondering, because right now we already validated cocaine-induced silent synapse in adult animals. So we are wondering how cocaine triggers the generation of silent synapses. Because synaptogenesis peaks at the early developmental stage, therefore, we looked for answer from literatures in developmental biology. We noticed that astrocytes actually play crucial role in synaptogenesis at an early developmental stage. Dr. Bambaris first discovered that astrocytes release factors called thrombus bondings. They can trigger synaptogenesis in nearby neurons. Surprisingly, those astrocyte-induced synapses, they only have NMDA receptors, but no empath. So they are silent synapses. Furthermore, Dr. Barry's elegant study also identified the neuronal receptor responding to such synaptogenic factor released from astrocytes. The neuronal receptor is alpha-2-31. It is an auxiliary subunit of an L-type calcium channel. So this alpha-2-31 located at the neurons, they serve as the anchor for the initiation of the synaptic formation. So the astrocyte release thrombus bonding. They bind to the neuronal alpha-2-31, and they together initiate the formation of a synapses. Inspired by his study, we hypothesized that in the nuclear accumbens of adult animals, cocaine triggers the generation of silent synapses via this astrocyte-mediated mechanism. So to test this hypothesis, I first want to test whether cocaine can activate astrocyte in the nuclear accumbens. Because astrocytes, they don't generate action potential. Rather, information sensing and signaling output from astrocyte is mediated by their intracellular calcium transient. Therefore, I took advantage of calcium imaging, and I used calcium transients as the functional readout of astrocyte activity. First, I expressed GCAM6 only in astrocytes in the nuclear accumbens of adult mice. Then I exposed the acutely prepared slice to cocaine local perfusion. I recorded the calcium transient in astrocyte in the nuclear accumbens before and during cocaine treatment. So my ex vivo calcium imaging showed that acute cocaine exposure significantly increased the frequency, amplitude, and the area size of calcium transients in astrocytes in the nucleocumulus, suggesting that acute cocaine exposure significantly stimulate astrocytes in the nucleocumulus. Then my next question is, if I suppress the astrocytes activity in the nucleocumulus, can I prevent the cocaine-induced generation of silent synapses? So to do that, I express a plasma membrane calcium extrusion pump called PMCA, specifically in astrocytes in the nucleocumulus. Then I train those animals to self-administer cocaine. And following 7 days of cocaine self-administration, I measure the ratio of silent synapse in neurons surrounded by astrocytes expressing the PMCA. In control group, the astrocytes only express fluorescent protein. My studies show that cocaine generates a considerate level of silent synapse in control group, but in the group receives the overexpression of a PMCA in the astrocyte, cocaine failed to increase the level of silent synapses. So my studies show that the astrocyte is activated by cocaine, and such activity is necessary for the generation of a silent synapse in response to cocaine. So here's a summary. The astrocyte, they release thrombus bonding 2, which gonna hooked up with the ARF231 expressed from both presynaptic and postsynaptic site from the neurons to trigger the formation of silent synapses. Importantly, if we shut down the TSP2 expression from astrocyte or ARF231 expression at either pre or postsynaptic neuron, or interrupt the binding between TSP2 and ARF231 subunit, we can prevent cocaine-induced synaptogenesis. In this paper, I further validated the key factors in astrocyte, and they can be targeted to attenuate chronic relapse of cocaine. If you are interested, you can check the cover article of the February 21st issue of Biological Psychiatry. So during the withdrawal from repeated cocaine exposure, cocaine first triggered the silent synapses. And those silent synapses, they recruit AMPA receptor and they mature into regular synapses. This newly added synapses significantly strengthen this connection, carrying the information related to drug and drug-associated cues. By targeting this newly added synapses, drug-associated memory and relapse can be manipulated. My former postdoc mentor, Dr. Yendo at the University of Pittsburgh, he comprehensively elucidated the cocaine-induced silent synapse and how they contributed to drug memory and chronic relapse. So right now we know astrocytes mediated the generation of silent synapse. Since this silent synapse ultimately can mature into functional synapses and significantly contribute to drug addiction behavior, it is critical to elucidate the molecular mechanism underlying the functional maturation of the silent synapses. So I keep focusing on what's the driving force underlying the maturation of those cocaine-induced silent synapses. So to answer this question, we must understand the synaptic plasticity at the early phase of addiction when cocaine-triggered silent synapses just emerge. I trained animals to self-administer cocaine or saline for a week. And then on withdrawal day 1, I recorded synaptic response and excitability in the nucleocumulus. My data show that cocaine self-administration increased the level of NR2B subunit of NMDA receptor, which enriched in newly formed synapses. At the same time, the memory excitability of nucleocumulus neurons is significantly reduced. As you can see in the panel, they fire less action potentials. So I examined the action potential traces and noticed that in cocaine-exposed animals, nucleocumulus took longer to recover from the previous action potential due to an increase of the median after hyperpolarization, MAHP. As you can see here, the cocaine actually, the trace take longer to recover. the small conductance voltage gated potassium channel, SK2, contributed significantly to the amplitude of the MHP. A pioneer study from Dr. Masago Ishikawa showed that suppressing SK2 channel restored the MHP in cocaine-injected animals. So inspired by his study, I want to test whether SK2 mediated cocaine-induced dampening of membrane excitability in the nuclear combus. To test that, I injected AAV expressing RNAi targeting SK2 messenger RNA in the nuclear combus. Then I trained these animals to self-administer cocaine or saline. As you can see, RNAi mediated SK2 knockdown prevented cocaine-induced increase of the MHP and the reduction of the membrane excitability. However, the control neurons, which are not infected by the AAV SK2 RNAi, still develop the hypoexcitability. So far, my data demonstrated that cocaine exposure increased SK2 to dampen the membrane excitability in the nuclear combus, leading to neuronal hypofunction. And other results show that certain synapses can recruit AMPA receptor to boost excitatory input during long-term withdrawal from cocaine. Therefore, I hypothesize that the dampening of membrane excitability acts as the driving force for the recruitment of AMPA receptor during long-term cocaine withdrawal. So to test this hypothesis, I will normalize membrane excitability after cocaine by preventing the upregulation of SK2 in a portion of neurons in the nuclear combus using SK2 RNAi. As you can see in the control cell, which does not have SK2 knockdown, the membrane excitability will reduce. And based on my hypothesis, this reduced neuronal function will drive the insertion of AMPA receptor to enhance the synaptic response. However, if I normalize the excitability change at the first place on day one, I remove the driving force for the insertion of AMPA receptor, so nothing will change. So if I compare those two groups of neurons with distinctive neuroplasticity at withdrawal day 45, I can test whether the membrane excitability at early stage serve as the driving force for the long-term neuroplasticity. And to do that, I need to do the dual patch. I predicted in those non-infected neurons after cocaine, the synaptic response will be increased. However, in those neurons infected by the AAV SK2 RNAi, because the excitability is normalized, there will be no long-term insertion of AMPA receptor. So if I compare with, between those pair of neurons, the non-infected one should always have bigger synaptic response amplitude than the infected one. So I conducted this dual patch. So first I inject the virus into the nuclear combus. Then I train those animals to self-administer cocaine. Then I subjected them to forced abstinence for 45 days. Then on withdrawal day 45, I conduct the dual patch from pairs of neurons, one infected by RNAi, the other not. So the pair of neurons, they receive the same stimulation during the dual patch recording. And this method allows me to accurately compare synaptic responses in these two adjacent neurons, developing distinctive neuroplasticity. Yeah, this process is very challenging to recording the closely adjacent neurons, especially those animals that go through the surgery, the training, the withdrawal. It's a very long experiment. And my results show that a neuron with SK2 knockdown at the early stage, they always exhibit significantly lower synaptic response than their non-infected neighbors to the same stimulation. Importantly, this reduction of synaptic response is not due to the side effect of the viral infection, because in the control group, for example, the saline-treated animals on withdrawal day 45, or in cocaine-treated group on withdrawal day 1, such a differences was not observed. So this finding validated that my hypothesis that the dampened membrane excitability drove the recruitment of AMPA receptor into the synapses during long-term cocaine withdrawal. So we're wondering, can we translate our synaptic finding to treat the drug addiction behavior? So we try to test the function of these factors critical to cocaine-induced neuroplasticity in addiction-related behaviors. I focus on chronic relapse, and I use a well-established behavior model called craving incubation. The craving incubation refers to the increase of the drug seeking, the craving, over prolonged abstinence. The animal trained to self-administer cocaine will press the lever even in the absence of cocaine, many times more on withdrawal day 45 than on withdrawal day 1. And I use AAV RNAi to knock down either NR2B subunit of NMDA receptor or SK2 in the neurons in the nuclear commons. Both gene manipulations curb drug seeking on withdrawal day 45, but not on withdrawal day 1. As you can see, the active lever pricing on withdrawal day 45 is significantly reduced in the group received that gene manipulation. So that suggests that these two factors are necessary for the neuroplasticity during prolonged abstinence, and those plasticity contributes to chronic cocaine relapse. So the summary 2, my studies show that cocaine exposure induced the generation of a silent synapse. And with astrocytes involvement, the silent synapse is triggered. Then the dampened neural excitability act as the driving force to facilitate the maturation of the silent synapse and strengthen the synaptic connectivity mediated the drug-related information. Therefore, those newly added synapses act as the neural substrate underlying chronic relapse to cocaine. So that's my study in my previous postdoc training. Since I joined Dr. Paul Kenny's lab at Mount Sinai, I focus on the function of the media venula to the interpeduncular nucleus in drug abuse. The illustration here shows the unique section angle, which can preserve the entire media venula to the IPN circuitry. The fluorescence image shows the expression of a channel rhodopsin in the IPN projecting media venula cholinergic neuron. I achieved this using retrograde AAV virus in cholinergic Chat-Cree mice. So this way allow me to study the synaptic regulation in the media venula to the from the media venula to the IPN circuit, and I can expose those mice to different drug treatment and measure the drug's influence on those synaptic plasticity. So this is a sagittal section showing the IPN. I circled IPN in the blue rectangle. The structure of the IPN creates extra challenge to brain slice preparation. Because IPN is at the ventral border of the midbrain, it is connected to the midbrain only through the fasciculus retroflexus, the axon bundle, entering the IPN at its caudal end. If we cut the slice chronally without adequate support or stable section, the rostral 1/3 or half of the IPN will easily disattach from the midbrain and get lost during the cutting. As you can see, the IPN is quite tiny. Usually a day, I only can obtain 2 or 3 slices. So I really cannot afford losing one of them. Therefore, the advantageous design of a compressome and the agarose protection tube are very helpful when I'm collecting the IPN brain slice. Also, the IPN neurons are highly sensitive to hypoxia. So the prompt sectioning will improve neural quality. The compressome allow me to cut the brain slice much faster than other vibratomes. And the compressome significantly facilitates our electrophysiology experiments. We also use the products from Scientifica. We really appreciate your excellent product and service. See, that's a zoom in image. If you cut the IPN without too much support, the edge of it, the rostral IPN will get lost. So I adopt the protocol of acute slice preparation in ex vivo electrophysiology recording into tissue collection for single cell messenger RNA sequencing. So rather than dissect out the brain and use the brain matrix to trim the slice and make the punch, right now we use the IC code NMDG cutting solution first to perfuse the animal, and we can cool down the brain to reduce the neural activity such so we can prolong the survival of those cells in the brain. And this electrophysiology standard brain tissue preparation method significantly improve the single cell RNA sequencing data quality. As you can see in those clustering graph, my method achieved more refined clustering resolution and specific detection of cell type transcripts, suggests my new procedure reduced tissue degradation and the resulting ambient RNA contamination. Yeah, and this picture showed me working in the machine shop, building some gadget for our electrophysiology or surgical stuff. And the photo shows the Mount Sinai School of Medicine. The black building here is our research center. And for the postdoc in our medical school, the first 3 years, the medical school give you subsidiary housing, which is right here. So usually when I go to work, rather than working directly to my office, I first wander into the park, just work like 10 minutes. Then I enter the building from the other side. Okay, thank you so much for giving me this opportunity to share my electrophysiology finding. And I can answer your question right now. Thank you.
That's wonderful. Thank you so much, Dr. Wang. And so we just had a quick comment by Chris, who found your experiments really impressive because the IPN is not easy to access. And now what I want to do is open up the floor and see if anybody has questions. Please feel free to unmute yourself or show your video and let us know.
All right, thank you.
Yes, we have a video coming out from Dr. Fernando von Hoff.
Yeah, we attended the same grade school.
Oh my gosh, that's wonderful.
Nice to see you.
Yeah, hello. Thank you so much, and great work, Dr. Wang. And you know, it's very impressive work. I do have 2 questions. Yep. First is about, you know, the silent synapses you were talking about. And my question is if you see differences during development, if younger animals show more of the synapses, and if you think they are involved in this process that is called synaptic pruning, where a lot of synapses are formed and eliminated, if you know anything about, you know, silent synapses and pruning during development.
Yeah, during the development, I don't know too much, but even in adulthood, during the drug treatment, there is a synaptic pruning. My colleagues show that the cocaine exposure, they increase silent synapse. And the morphine treatment also increase silent synapse. But morphine-induced silent synapse is opposite. They cause the internalization of AMPA receptors. So they turn the originally functional synapse into silent synapse. Then after those synapses become silent synapse, they become very fragile. Then maybe the microglia around them can pruning them. So after morphine, the total spine density actually reduced. And because the regular synapse, first they downgrade into silent synapse. They don't have AMPA. Without AMPA, they become even more unstable. Then they just disappear. So silent synapse could be a transition between the stable synapse and the no synapse. It's very hard to know whether they grow out or they just disappear from the regular synapses.
I see. Okay, great. Yeah, thank you. That's yeah, very interesting. And the second question was, you saw the astrocytes show a response to cocaine.
Yeah.
And my question was whether you think cocaine was acting directly on the astrocytes, or if you think is acting on the neurons and the neurons are activating the astrocytes. So what is your
Yeah, because actually the neuron and astrocyte, they share the same as sister cells, right? So actually all the receptor neuron has, astrocyte also have. So astrocyte, they have a dopamine receptor. Cocaine binding to the dopamine transporter increase extracellular the dopamine. So our hypothesis is actually, I think some paper already tested that. They block the dopamine receptor. They just apply the general blocker on the brain slice. So they block both the neuron and astrocyte dopamine receptor. And when you apply drug, which triggers the dopamine release, it no longer can activate astrocyte.
I see.
Yeah.
And has anyone done the blockade of only the astrocytes, dopamine receptors on the astrocytes and not the neuron?
That's very hard because they are the same. The only way to do it, maybe you do a astrocyte specific knockdown. Right. Another tricky thing is right now, because in neuron, a lot of people, they use DRAG. But if you use that, you use the DRAG, the GI coupled signaling cascade, try to suppress the astrocyte activity. You cannot. They have an initial very short lasting reduction. Then the astrocyte are gonna compensate and recover from that. So the DRAG does not work in astrocyte. So there's no way you can effectively and manipulate astrocyte activity. I think that's the bottleneck for the field because we try to manipulate the astrocyte activity to test the behavior change. But right now there is no very powerful and accurate tool to do that.
I see. Okay. Awesome. Well, thank you so much for your talk. And thank you for the organizer for this great talk.
Wonderful. Thank you for your question, Fernando. I want to keep opening it up and see if anybody else has any questions. Please just feel free to unmute yourself and shout out or turn on your camera and I'll call on you. Give it just a couple more seconds. If not, then please feel free to contact us at info@precisionary.com if you have questions later on, or if you think of something else to say or comment or like some more information, we will be sure to respond. And with that, I'd like to thank Dr. Wang for your time and for all of our attendees for being here. And with that, I wish everyone a wonderful week.
Thank you. Bye-bye.